very closely with Rhiannon is Kathleen Keatley, who's a first-year PhD student. So she's only been in post for a very few months, but she's already well underway with her project looking at glioblastoma and looking at these mitochondrial defects, which might be a very good area to investigate for new therapies for glioblastoma. Kathleen?
Well, the project I'm working on is titled The Effect of Mitochondrial Mutations on Glioblastoma Multiforme. And as Rhiannon mentioned before, we're looking at alterations to these mitochondria to see if they have any effect on the brain tumours. And we're doing this in a joint fashion, looking at the mutations structurally to help create a map of what those mutations are doing, and then hopefully looking at them in the lab, see if they have any impact and any effect on the cells we're working on.
So the long-term goal?
Well, hopefully, because the different cell lines we're working on have different subsets of alterations, if we can look at how these affect the treatment protocols or response to different treatments, it might in the future tailor the treatment plan towards the patient.
And how have the first 6 months or so gone? Are there any major findings that you think are exciting?
Well, we found that the cell lines we're working on have quite a number of alterations, which is not what we expected, but it gives us a lot to work on, so it's been going good.
Okay, I think that'll probably be, but it doesn't really matter about that. So the other project that we have, which is supported by Headcase, is a project on something called Gas6-axle signaling pathway, and this is involving Dr. Sasan Hafizi, and he's co-supervising Mikaela Vowry. Mikaela is a little bit further down the line in her PhD. She started last October, but she's working on this particular signaling pathway, and Mikaela will tell us a little bit about how this project is going and what the main aims are.
So we're working, as Professor Jeffrey Pilkington said, on Gas6-axle signaling pathway in GBM cells. These receptors have been found to be involved in the invasion and proliferation of brain cancer cells. So our main focus is trying to identify their function and the signaling event downstream that aid the tumorigenic effect in glioblastoma multiforme, and trying to block this effect by using small molecules inhibitors, which can be further used into the clinic later on in combination with standard treatments for brain cancer patients.